Okay, in this view of purine and pyrimidine nucleotide bases and their biosynthesis, I'm going to be distilling down the essential elements into something that is manageable and understandable. The biosynthetic pathway of purines and pyrimidines is a bit complicated, and we are just going to focus on the large intermediates that mediate the key reaction steps. So for purine biosynthesis, let's start there. The purines, of course, have the general ring structure like this. So we have to synthesize this general ring structure. But in order to do that, we are going to start out with the substrate, ribose 5-phosphate. Recall, this is a product of the pentose phosphate pathway. The pentose phosphate pathway is involved in NADPH generation, but also in the non-oxidative pathway is responsible for our 5-carbon sugars for ribose 5-phosphate for nucleotide synthesis. Now recall that if we do not need ribose 5-phosphate, these are returned to glycolysis in the form of fructose 6-phosphate and glyceraldehyde 3-phosphate. This is essentially to capitalize on these carbons being potentially oxidized into energy in the glycolytic pathway. Now, if we need ribose 5-phosphate, we will proceed down a path of generating PRPP. So this step is mediated by PRPP synthase. PRPP stands for 5-phosphoribosyl 1-pyrophosphate. Essentially what we are doing is maintaining the 5-phosphate group on ribose, which is going to be used for linking nucleotides in the DNA and RNA synthesis process, but we are also now adding phosphate, pyrophosphate, 2 phosphates on the number 1 carbon. What this allows us to do is to charge the molecule. So this is an energy utilization step. And so this is a one-way reaction. This is not reversible. So not being reversible, being an irreversible reaction, this is a great place for inhibition and regulation. Now for purine, we'll see this in a second, but for purine biosynthesis, what we're going to do is we are going to start out with our ribose sugar, and we are going to have our phosphate group on here. This, of course, is our number 1 carbon. Here are our hydroxyl groups on the number 2 and number 3 carbons of the ribose structure. And then, of course, on our number 5 carbon, we have our other phosphate group. So this phosphate will give us our 5' to 3' linkage when we link up these nucleotides into nucleic acid base structures, DNA and RNA. And then this pyrophosphate group is going to drive the reaction forward. So this is PRPP. Now for synthesis of purines, we are going to synthesize the purines on the number 1 carbon and begin the reaction by hydrolyzing these phosphates group. So ultimately we will have a purine attached to this portion of the ribose ring. in this fashion. Once we assemble the ring using the platform of the ribose structure, we will have a product that is called inosine monophosphate, abbreviated IMP. Through a series of reactions, we can generate from IMP either AMP, adenosine monophosphate, or GMP for guanine monophosphate. Adenosine or guanosine monophosphate. Now let's go back up here to the regulation step of PRPP. These are regulated by purines. For example, AMP or GMP. They inhibit the PRPP synthase enzyme. So if we have sufficient amounts of our purine nucleotides, they will feed back on PRPP and inhibit the enzyme. This will block the entrance of ribose 5-phosphate into the nucleotide synthesis pathway and shunt it into glycolysis to take advantage of those carbons. As our levels of AMP and GMP begin to decline, now we want to synthesize purines, so we allow PRPP product to be formed. We build the ring structure on the ribose, generating IMP, and then modifying the ring structures to give us AMP or GMP, adenosine monophosphate or guanosine monophosphate. Now for pyrimidine biosynthesis, we have a slightly different path. Here, we are going to synthesize the ring structure without the ribose. So by attaching the ring structure first, we can then add it to PRPP once the ring is formed. At this point, what we are constructing is a molecule called orotate. And this, of course, happens by several steps using amino acids and small molecules such as carbamoyl phosphate to close up the ring. Now once we have orotate, at this point we can add it to PRPP. With additional processing of this structure, we now have uridine monophosphate, or UMP. The UMP then undergoes reactions and several processings to generate CMP and TMP, cytosine monophosphate and thymidine monophosphate. There is a particular block in this reaction. Up here, when we add the orotate to the PRPP, this enzyme is called orotate phosphoribosyl transferase. And a genetic error in this process here of adding the orotate to the PRPP will lead to orotate aciduria. So this is a genetic inborn error blocking the coupling of orotate to PRPP, and thus we have excess buildup of orotate, which is an acid due to this carboxylic acid, leading to aciduria.